everyone, welcome to Shoals Marine Lab. I'm Matthew Lynn, and today we have with us Collin, our crab parasite intern.
Hi there.
Let's get started. So Collin, let's start it right off. From where are you?
I'm from Grand Rapids, Michigan, and I go to school at Michigan State University. I am a senior studying fisheries and wildlife, and this is my first summer here on Appledore Island.
And where are you right now in your career? What do you hope to do next?
After I get my bachelor's, I hope to take a year off and do some field work, just kind of get an idea of what real field work is like. Also to give myself a little bit of a break from academics, and then after that year off, I plan to go to graduate school. So in that year off, I'll definitely be looking into different programs throughout the country and get an idea of what advisor would work best for me, and developing a research question with them, and then getting my master's.
And what got you inspired to start a career in science?
Since I was ever a little kid, I was always interested in anything outdoors. I was always playing in the creek, catching frogs and turtles, trying to bring snakes inside, and that quickly led me to begin volunteering at an exotic pet shop. From then, after 2 years of volunteering there, I started working there and realized that I needed to do something with the environmental science field, and that's why I chose to go to Michigan State, because they had such a great research opportunity and program for environmental science.
And moving on to our technical narrative, why would you say that crab parasites are important to the audience?
Yeah, so I believe that parasites are extremely important because they make up roughly 30% of the identified species on this planet. So what we do know exists, at least almost a third of them are parasitic in nature. So they make up a large part of our ecosystems, therefore they have a large impact on the population interactions of different species. Now, Carcinus manus is an invasive species that came here roughly 200 years ago on the hull of boats, and since it's been here, it's had some significantly negative impacts on some native biota. So some people may be aware the cancer crab is a commercially valuable species here in the Gulf of Maine, and it has been pushed subtidally because of the presence of Carcinus manus. So by studying parasites and how parasites are impacting the populations of Carcinus manus, we can better determine if these impacts on local populations that are being pushed out of their native range are going to be long-term, or if they have possibility of bouncing back.
So you've convinced me that parasites are important. What is the question you're trying to answer?
Yeah, so I'm looking at whether or not parasites are causing altered predation rates of green crabs. So my specific question is whether or not parasitism is causing an increase in predation rates by gulls of Carcinus manus, the green crab. And my prediction is that infected individuals that are exposed to the parasite will be subject to increased predation versus uninfected individuals. And this is partly what I believe to be due to sluggish behaviors that are exhibited after initial infection. But I'm not specifically attacking the why of that question. So why would I see increased predation? I'm just trying to determine whether or not that trend does exist. And hopefully my information that I provide in my experiments will act as a bridge for further experiments.
And what materials and methods are you using to answer this question?
So to begin my experiment, I'll be running exposure trials for 72 hours. I will have a control tank and an exposure tank. In my control tank, there will be no parasites, and then in my exposure tank, there will be parasites. And after I run that 72-hour period of exposure, I'll be tying both the control and the exposed crabs out in the intertidal via tethering experiments. Tethering experiments basically require an organism to be attached to a line and then anchored out in some site. And so I'm doing it in the lower tidal zone where the green crabs are typically found, and then after a 24-hour period and a 48-hour period, I'll be going out there and counting how many individuals have lived and how many individuals have been consumed in order to get an idea of a measure of predation rates between the exposed and unexposed crabs.
And what have you found so far with these tethering experiments?
So as of today, I've run 2 tethering trials out in the intertidal. With my first trial, I found that there are actually more unexposed crabs consumed versus exposed, which is really interesting and the opposite of my predictions. So there's definitely some analysis that needs to be done there. And then with my second trial experiment, I have found after 24 hours that only 1 crab was eaten. But I believe that it's in part due to the fact that yesterday we had a very large storm roll through, and perhaps gulls were not foraging during those periods of time, and that's why I'm running 5 replicates in order to account for any kind of noise like that in my experiments. And then going forward, I just started my next exposure trial, and I'll be tying those guys out in the intertidal in 3 days, and hopefully from there I'll see some trends that are consistent.
Well, thank you, Collin. That's about all the time we have for today. Today we got to hear about the wonderful crab parasite internship. I'm Matthew Lynn, and we'll see you next time.